Yes, Arya, you can start now.
Yes. Good evening, everyone. A warm welcome to all the participants. On behalf of the Microbiology Society India, I welcome you all to today's guest lecture session. Today, we are privileged to have with us Dr. Nagabhishek along with his team from Imperial Life Sciences. Dr. Nagabhishek is currently working as an application scientist at Imperial Life Sciences. Today's session will focus on advanced super resolution microscopy technologies from ONI and their application in life sciences research. We are delighted to have Sir and his team with us today, and we look forward to an informative session. Without further delay, I request Dr. Nagabhishek sir to kindly begin the session. Thank you.
Yes, Prathamesh, have you started recording? Prathamesh.
Yes, sir, started.
Okay, thank you.
Yeah, thank you one and all for joining for this meet. And I would like to thank Deshmukh sir to provide this platform for us for presentation on super resolution. And I welcome one and all for this talk. And let's start with the talk. So today I'll be speaking about single molecule light microscope or single molecule localization microscopy. So I know like this is a microbiology society and you people are working a lot on confocal imaging and things like that to get your accurate imaging things. But because whatever you're looking at, it is at a nano scale, so it is very difficult to image, and because of the diffraction limit what the confocal or the STED microscope offers. So for this questions or the difficulties what you're facing, so that would be resolved using a super resolution microscopy, and that's what we are showing or demonstrating things today. So we have recently channel partner with ONI. ONI is a UK-based company which specializes in single molecule light microscope. So most of the microscopes are very comprehensive and very big as such. Like our microscope is very compact and it can be put on any table. You don't need dark rooms, and it is very much user friendly. Any student wants to use this microscope because it's go-to. So it is very small, compact, and it is very easy to use, and that's what the ONI specializes or USP is all about. So why we need a single molecule or single molecule particle tracking, or how it can be used for microbiology and EVs research, we'll just go through one by one. So I'll just start with part one, which shows about our introduction for ONI. So we just completed 10th year anniversary for 2 days back, and so we are a decade old company now. And if you see in the map here, we had our first installation in India. We just channel partner with ONI. It's been like 2.5 to 3 months now, and we had our first installation in Aizer, Bhopal now. So that's a very good thing to start with. And we have got a couple more inquiries, and we are just very active in the market now. And to see on a global perspective, we have 330 plus installations. We have more in academia and in biopharma. It's a mixed ratio. So we have 180 plus installation in academia, and we have around 100 plus installations in pharma also. So we are having an equal balance of both of it. And if you see in academia, we have put it in top-notch universities like Stanford, Oxford, Harvard, Penn State, and NIH and cancer research in UK, and so on and so forth. And if you see in biotech or pharma companies, like most of the companies like Merck, AstraZeneca, Novartis, Roche, everyone owns or have one system of ours. So majorly, if you see people who are working on EVs, people who are working on virology, people who are working on bacteria, they are the right clientele for us. And even in case of biopharma, like a lot of people who are working on vaccine development and LNPs, so lipid nanoparticles. So these are the people who ideally use single molecule light microscopes to evaluate or to characterize their study as such. So where do we stand as a single molecule localization microscopy? If you see the conventional microscopes, so it can resolve up to 1 micrometer in size. So, and when you come to higher resolution microscope like confocal or SIM, which you generally use on a regular basis, so the diffraction limit, what you can see is up to 250 nanometers is what you can image. Like the highest, the low end resolution, what you can touch is around 250 nanometers. But in case some people would have also worked on stud here. So stud microscopes allows you to go down below that. It will achieve 70 nanometers of resolution as such. And what we are speaking today is of SMLM category. That is where the ONI comes into picture. So who are the people who uses this? People who are working on at a single cell level, people who are working on like bacteria, people who are working on virus, people who are working on EVs, people who are working on antibody characterizations. These are the right set of people to use this super resolution microscope. And the next category comes under mini flux, where up to 1 nanometer can be seen. And in case of our ONI, so most of the SMLM will be resolving up to 40 nanometers, but in our case, we have shown up to 10 nanometers, and in fact, up to 6 nanometers, but we claim it as 20 nanometer resolution, but people have shown through publication up to 6 nanometers. So that is what the resolution limit it can go down to. And if you see for electron microscope, it comes in Armstrong as such. So we'll just focus on SMLM today and our ONI today, and how we can do, and what are the things that we can exactly do as such. So the key applications for people who work in this domain is cytoskeleton research, molecule and cell characterization, then EV profiling, then how cargoes are loaded, not loaded, and if you want to study the endocytosis of EVs and exocytosis of these EVs, or you want to study the endocytosis of a receptor. So all these things, or virus protein interaction. So all these kind of studies can be seen. So I have kept my presentation morely focused on virology and microbiology things today, and I'll also show you what all you can do in single cell also, which helps, or you can plan your experiment later on in your future if you're thinking about SMLM. So I'll just give you a brief about what all it can do, and also specifically concentrate later on virology and bacteriology as such. So the core applications, like people who we have classified under 4 segments who use our microscopes exclusively. More than 60 to 70% people use for EVs and LNPs, and 10% people use for pathogens, and neurosciences for synaptic complex or synapses studies. They are using it, and people have started using exclusively for CAR-T interactional studies, and so on and so forth. So immuno-oncology has been picking up off late. So why only? There are several other companies which can give a single molecule localization, but none of them give a complete ecosystem or a suite of solutions. So here we start with, like, in SMLM, everyone gets scared with the technique because it is really hard to standardize it. So you don't have to worry about that anymore. We have given you ready-made kits to do that. So you can start your SMLM right now on the bench, and you can get your result on the first go. That simple is what we have made sure that you can get our kits, and it's an open system. We don't recommend you to buy our kits. It is absolutely an open system. You can use whatever you want. We don't pressurize you to buy our kits or consumables as such. So, but yes, we do provide kits, and we do provide the software analysis. So in most of them can do SMLM, and after that they struggle with the analysis. They use multiple pipelines, multiple tools to do that. So we have simplified everything. We have created a Cody software, which is a cloud-based software, and every pipeline you can be put in that, and analysis is made very simple, step after the step after the step. So within 10 minutes, you will be able to learn that software and get the complete analysis, and it is publicationable data would be ready for the things like that. So coming to our scopes, we have 2 scopes. One is called Nano Imager and one is called Apollo scope. And this is our Apollo flow system, which is an automated solution system where if you're using a regular protocol in the lab, you can automate up to 20 slides. It is an automated staining solution station, what we offer. And this is how compact the system is. So it is just, you can imagine a size of a A4 size sheet. That is the breadth of it, and length is almost 3/4 of the A4 sheet. That's small, and that's how compact it is. And this is the only system dedicated for SMLM which is so compact. So currently, if you see all the traditional SMLM labs, it will look something like this, because the systems are very bulky. You need a complete room infrastructure. You need a dedicated manpower to get trained on the system, and so you need a dark room facility. You need a vibration-free table. There's so much of infrastructure cost involved to set up a microscope. But now you can just put this microscope on bench and you're good to go. So whatever you see here, this is the scope. You don't have traditional eyepiece and so on and so forth. So you just have a cool laptop, and we also give you a joystick to play around, to move around. So it is as simple as that. You don't have to worry about the complex settings, because it is a pre-calibrated instrument. And this is a laser box, what you see here. So this is the only laser box what you have to. It is the size of a UPS. CPU. So that's how the small it is. So as I was speaking about the ease to use, so you can start using your imaging very faster, because other SMLM system takes really long time to adjust your optics, adjust your laser, to calibrate your lasers, and so on and so forth. Here, you just switch on the microscope, you're done, you're ready to go, because it is all auto-calibrated. Lasers are pre-aligned. You don't have to worry about anything of that sort. And it is very flexible with illumination modes. So people who work on SMLM use different modes like high-low mode, turf mode, and so on and so forth. Here you can manually change the modes of angulation of laser. It will help you to put down or to get your focus in a more flexible fashion. So we also do fixed cells, and we also give you an option to do live cell. That is good. So you can put your CO2 cylinder connected to the microscope. You can image for 24 hours or 48 hours, and so on and so forth. Even our programs are flexible. That means to say, even if you want to create your own pipeline, where in our software, if there is no solution for your analysis, and you want to create your own pipeline, yes, you can write your own Python code, and you can do a plugin to our software. Our software team will help you to integrate your Python code into the software. So that's how flexible we are in creating your own pipelines also. And we are a class 1 laser. That means to say that there is no laser leakage. You don't have to undergo any laser training for this, or you don't have to follow any laser protocols. You can put this machine anywhere, and you're good to go, because it is coming under class 1 laser. So biosafety compatible, that means to say, like people who work on virus and bacteria, they cannot, sometimes the core will not allow them to take their samples. So it is very difficult for them, or they cannot move from the BSL-3 or BSL-4 facility. So even we have certain companies and certain labs worldwide, where they have put the system inside the laminar air hood. So they are taking images there only, so that they don't contaminate the samples of BSL-3 and BSL-4. So to give you a bird's eye view, this is what the modes we offer. That is high-low, turf, and epifluorescence is what the microscope can do. It is a 4 laser system, that is 405, 488, 560, and 640. These are the 4 lasers, and you'll have a split screen. That means to say you can do 2 simultaneous imaging at once, and you don't need optical table or special room requirement, as I mentioned before. So what are the super resolution techniques? What people do is Storm, Palm, and DNA paint, and we support all these 3. And apart from that, we also support single molecule tracking, that is called SPT, and we also support SM-FRET. So only microscope which supports SPT with SM-FRET with all 3 is the only dedicated system from ONI. So this is what our another USP is. And we use most advanced CMOS camera here, so which is called Hamamatsu in case of our nano imager, and we use Teledine in case of our oscilloscope, which is more advanced. So, and one good part is you get a temperature control stage here. So that means to say, when you're doing live cell imaging, you can set your temperatures to 37 or 38 or 36, whatever temperatures you want to, and you can do live cell imaging. It is stable, and we give you humid chambers for it to do so. And another good part is you have autofocus mode. So once you come on the Z plane, you don't have to focus every time. So you can scroll around once the autofocus is locked. And So these are the major USPs of our imaging as such. So coming to part two, so I don't know how many people are already users of single molecule light microscopy here. So I just wanted to add a single slide to explain you how SMLM is different from other microscopes. So if you know that SMLM microscopes
अभी भी नहीं
like
लाए
hello, yeah, someone has a question. Okay. So if already people are using confocal microscopes, right? So in case of confocal microscope, you use a pinhole and you illuminate the laser through that, and you get your imaging done. And people who work on stud, it is more of shrinking the spot, making the spot concise. So that's how you get a super res imaging. So in case of SMLM, we do little different. We makes the molecule blink, so each and every molecule is made it to blink. So that's how you'll be able to capture. So it's like, to just keep you a layman scenario, so if you're in a group of 100 people in a room, so if I tell you to switch on the light from your mobile, so everyone starts switching on the light. So if all are switched on the light at once, we cannot know the intensity of light, or we cannot identify single light from whose phone it is coming. But if I ask you to do on a random fashion, now I'll be able to identify from whose phone the light is coming. So it is very similar to that. So the SMLM core concept is you can use SMLM when you're able to make your fluorochromes or proteins to blink. So that is the main thing. So you have to have a blinking capacity of fluorochrome. So each emitting molecule appears as a diffraction spot limit, and we use a Gaussian fit theory to fit the exact molecule there. And the photon count matters a lot. So better photon count, that means better intensity providing fluorochrome should be used. And because we repeat this process for 10 to 50,000 times to get the super resolution image. So your size of the files are really big, but the image clarity what you get will be the best. So that is what you do in SMLM. So typically 10 to 20 nanometer resolution easily you will be able to achieve using SMLM. So as I said, we have different modes: epifluorescence, high-low, turf, and life cell imaging. So people go for different modes based upon their usage. So you have to understand two things here. One is the modes, and second thing is the type of super res imaging technique that you need to adopt. So it is either DStorm, PALM, or DNA paint. So having said this, so let me give you brief glimpse about each and every mode and what and when it is used. So epifluorescence mode is generally used when you want the entire cell imaging, or when you are trying to look at bigger scale events. So high-low is highly inclined and laminated optical sheet, which means to say that your lasers are inclined to cytoplasm level. That's it. So when you're looking for cell organelles like ER for Golgi, and you just want to focus on the organelles inside it, generally people put on high-low mode. So this reduces the background noise and so on and so forth, right? And there is one more mode like called TURF. It is a total internal reflection. So where completely the laser bounce backs from the cover slip. So people who wants to study the membrane receptors, so trying to resolve only the membrane structures, membrane interactions, and so on and so forth, people usually put on TURF mode to get a right illumination. So once you're fixed up on your modes, then you'll be coming to the super resolution microscopy. Like there are, as I said, there is DStorm, PALM, and paint. So to give you a brief about what we just said, like we spoke about SMLM, but we wanted to understand exactly how this SMLM works and how the Abbe limit or the diffraction limit is being squashed out. So to understand that, I've just put this slide. So usually your GFP proteins considered as it is 2.5 nanometer, but the signal spot what you get in confocal is around 250 to 400 nanometers in size. So, but your single molecule localization, when you say if there is multiple GFPs, then you see a very big spot. So the way it is being resolved, as I said, it first identifies a molecule, and we apply a Gaussian fit curve, and we identify that this molecule is here, so we have localized it, and then we go for super resolution imaging that by taking multiple images. So suppose you want to understand some nanoscale events which are close to each other than the limit of resolution. That means to say that your GFP protein will glow back into more than 250 nanometers to 400 nanometer limit, and you will not be able to appreciate the nanoscale events. So this is an example to illustrate what I'm saying. So this is your Abbe limit, that is around 250 to 300 nanometers is the scale. And suppose you want to check this calcium channels. And so each one GFP protein will just show you this much of diffraction. So how will you able to see exactly the calcium channels or calcium entry and so on and so forth? So in case of every cell, there are like 1,000+ events going on. And if you want to understand the nano scale events or signaling what is exactly happening, you should have a better resolution, right? So for people who are working on that scale, so definitely needs a super res microscope to understand this. And that's what we have here to achieve that super resolution of 10 to 20 nanometers as such. So usually the Abbe limit, as I said, it is usually wavelength by lambda by 2. And with standard microscope, if I want to resolve these many molecules which are in a particular place, so it gives you a diffraction limit of 500 nanometers, and you can hardly see anything here, as you just see as a red spot. That's it. You will not be able to see what molecules in which positions it is being present. But in case of single molecule, you're making the molecule blink on a randomized fashion. So first you localize the molecule, and once you localize the molecule, you apply the Gaussian fit, and then you precise the molecule, and then you repeat the process for more than 5 to 10,000 or up to 50,000 frames to resolve it. So usually if you see under the microscope, like you see the blinking something like this. So you see the blinks, what is happening here. So each and every blink will be taken up, and the software localizes it, and whatever the process you saw on the previous slide will be done, and it reconstructs it back to give you a final image. So every filament you see, there is a dotted pattern. So each and every actin now is being coded. So every actin will be coded. That's why you see a very dotted pattern there. So each and every molecule of actin will be acquired here. So that's what it does. So this is one more example to just show you like how it happens. So STORM is one of the technique what we use for fixed samples. So here we are showing EVs as such. So in normal confocal microscope, if you're trying to take EVs, you'll fail to take EVs because it is very difficult to resolve the EVs as such. So once you stain the EVs and once you do it, if you zoom it also, it looks something like this. If you submit this to a publication saying that this is an EV and this is and so on, so forth, they will definitely not accept it because there is no clarity on imaging here. But in case of our super resolution microscope, see here, once you have stained your EVs, now you exactly know what are the types of receptors and what are the types of staining it is being done, and this is the amount of resolution what you try to get now. And this is very convincing for any reviewer to say that yes, in fact, this has got your 3 different receptors, and your whether your cargo is loaded, non-loaded, everything can be characterized using this. So yeah, moving on to a part of technique, as I said. So DSTOM and PALM are the most common super res techniques which is generally used in most of the labs. Nevertheless, DNA paint is also being used in most of the labs. So in case of a DSTOM, you can do up to 3 colors. In case of PALM, also you can do up to 2 to 3 colors. And in case of DNA paint, you can do up to 5 to 6 colors, or it is unlimited technically, we say. But to standardize DNA paint, it is little difficult. And we also offer you kits to do DNA paint, so you don't have to worry about that. We give you a microfluidic chip to do your DNA paint. So what is the major difference, and what are the techniques when you have to use what? So most of them gets confused when it comes to single molecule localization microscope. So as I mentioned before, DSTOM is a technique where you use for fixed samples. So when you're going for antibody, like similarly in case of confocal, how you're doing confocal, if you're doing in a similar method fashion, then you'll be using DSTOM for fixed samples. So you'll be using antibody, which is attached with the fluorochrome. And the fluorochrome has to, whatever the fluorochrome, what you're using, it should have a high intensity molecule, and it should also be able to blink. So these are the two criteria for it. And usually in DSTOM, in case of confocal, you just use 10 millivolt of laser power. But in case of DSTOM, you'll be using up to 100 to 150 milliwatt minimum of laser power to make the molecules blink. So if you ask me, like, what are the general molecules that can blink? All the Alexa series can blink. Most of the Atto dyes can blink. So it has got a self-blinking nature. So you can use these kind of fluorochromes to do that. And you need a specific buffer to do DSTOM, and this is really important. Why it is important? These buffers, when you add, so it will make it it will enhance the photo bleachability property. You can do imaging for a lot more frames. So your buffers are really important, and we also provide you the buffer solution, so you don't have to worry about that. And or you can prepare your own buffers and you can try out. So there's no compulsion that you have to buy buffers from us. So yeah, this is the main thing with DSTOM. Coming to PALM is usually done on live cells. Usually fluorescent proteins are used. Usually photoactivable GFPs or photoactivable RFP or M cherries can be used in case of PALM technique. So you usually try to transduce or transduct the viral particles to make it make the cells prepared for your live cell imaging. And protein of your interest also can be tagged with one of these fluorescent molecules. So you prepare your cells in such way. It is a little lengthy process when you go for PALM as a technique, but you will have a wonderful result. And once you prepare something for your PhD or for your labs, that would be there forever. And from there on, you can do the imaging at a very faster pace because you have created the model system as such. So here, in case of PALM, we don't need buffers, and we don't use very high laser power because it is live cell. You don't want to kill the cell. So that is how it is being taken care. So I'll just run through some couple of slides where we have demonstrated or where we have showed for different applications as such using different super res techniques. So people who work on cell, exosome, lipid nanoparticles, or adenovirus or antibodies, these are the ideal people who use our things. So as I told, EVs are the main customers. And if you see most of the EVs in most of the primitive journals, you see the EV imaging something like this. So this is not accepted anymore in the case of EV fields. They want it to be precise. They want to know exactly how or what molecules have been stained and how, because even the characterization gets difficult if you want to characterize this clustering analysis, or if you want to count the number of EVs, or if you want to pull out single stain, double stain, or triple stain EVs, it gets difficult. So there is no clarity in this kind of imaging anymore. So people ask for high resolution imaging now, and that's where it comes into picture. Where you can show pretty good images as such. So this is one more technique. So whatever we showed here, it's a DSTOM based EV technique. So what I'm showing here, it's a PALM based technique where you're doing or visualizing a release of EVs as such. So it has been demonstrated in endocrine PC2 cells, and it has been these EVs have been tagged with CD63. So which is pH lurin. That means to say that once the EVs have been released and touch the membrane, the pH change happens, and only then it glows. So people who wanted to study the EV release, they can use this kind of technique to resolve their questions. And directly you can visualize the cellular release of EVs. You can monitor EV release through, if you want to do various stimulus and you want to check how the release is happening, you can do that. And you can also define the trafficking path. So complete every EV can be tracked. I will also show you that tracking analysis. So this is one more where we are showing with SPT. So atlastin is a protein. What in this study they wanted to understand the atlastin protein, which is a GTPase protein. So usually, which is present in the ER region. So the major function of this atlastin is to just keep the ER network intact. So they wanted to check on some addition of their inhibitor or anything, whether the atlastin activity was reduced, and to see such proteins and to also track like how exactly the movement of this atlastin molecule is going. They used this live cell with SPT as such. So, and another major people who take our microscopes are for characterization of LNPs. So, or virus-like particles, VLPs, we call it as. So we have a complete solution for characterizing LNPs, to run the analysis of LNPs and so on and so forth. So when it comes to LNPs, first you wanted to know whether your LNPs have been intact. So you do a pan LNP stain, and then you wanted to see whether your engineered ligand or protein of interest is been exactly attached. So you characterize that using one secondary primary antibody for that particular protein. And you wanted to also check whether it has been loaded or non-loaded. So these type of characterizations are mainly done in most of the labs, and most of the pharma which is used for vaccine developments use this is the major LNP characterization. So where we have showed a pan LNP marker here, and we have calculated the size, and we have also showed whether it is been loaded with RNA or not loaded with RNA encapsulation. And this is one of the study to show with DNA paint for super res imaging. So here we have used DNA JF646 Horseshat stain, and we have shown the membrane proteomic gold as a contrast thing. And this is how the DNA paint looks like when you do. So DNA paints are very crisp as such. It will give you a very beautiful image, and it is very intense. And if you try to optimize with DNA paint, that would be great. And because you can do multicolors in DNA paint, as I said. So in case of DNA paint, what happens is you just tag your fluorochrome to a DNA strand, and that DNA strand will be attached to your protein of interest. So that's a complementary strand, and that's binding and unbinding happens continuous fashion. That's how the blinking of these molecules is being determined. Once it attaches, it gives your fluorochrome. Once it detaches, it doesn't. So that's how you'll be able to visualize here in case of DNA paint. So this is one more where they have shown that EV uptake study they wanted to do. They've used the human umbilical endothelial cells here. So in case of these cells, they have tried to track each and every EVs, like one's they've taken the umbilical cell and they've added some isolated EVs from humans, and they wanted to see at what percentage EVs has been uptaken, at what time it is uptaken, and how much it is uptaken, and after the uptake, whether the EVs have released the cargo or in the cell or cytoplasm, or it went for a death pathway. So complete fate mapping can be done of every EV here. So that's what we have shown here. And there's one more study where PDZ8 protein. So usually, again, this is the ER protein which gets stabilized when it comes in contact with mitochondria. So it helps in calcium exchange and so on and so forth. We have used COS7 cells to demonstrate these pantas. So each and every panta, what you see here, shows you the active molecule of PDZ8, and it also shows the exact localization or touching to from ER to Golgi complex and so on. The tau proteins have been formed Am I audible? Did I get there was some connection issue?
Yes, yes, there was connection issue, but now you are audio.
Hello, Deshmukh sir?
Yes, now you are audible. Now you are audible.
Oh, okay, fine. So, yeah. So what I was uh, what I was showing you was about tau protein localization and so on and so forth. You can do such characterization studies. This is very important because you need to understand the pathology of tau protein, how exactly it interacts. So whether branched tau protein fibrils are formed or single tau protein fibrils are formed. So understanding this kind of information will help you for disease progression and prevent the aggregation of these molecules. So you can also do one such from it is a demonstration of another FFP slide where ER is being stained for breast cancer cells. This is a negative control, non-cancer control, and cancer control. So yes, you can do a lot of application on using tissues. You can use microorganisms, yeast, bacteria, virus, so on and so forth. And one more major application of our thing is SMFRET, and SMFRET people who work on SMFRET people are relatively small, but people who use SMFRET, they love our system because SMFRET in short to say you that you're trying to understand whether the conformational change is present in the protein or not. Like if your ligand is binding to your active pocket, if you want to check that, you want to validate that, SMFRET experiments will help you to do that. So in the case of layman understanding, what you try to do here is there is two molecules here. So you attach two molecules at 10 nanometer difference, which is an acceptor and which is a donor. So it is green is an donor and red is an acceptor. So usually what happens is when you excite a laser to green, so automatically because the energy transfer is there, it will come in red channel. So that means to say that still there is an intact positioning and there is no conformational change happen. But when your molecule is bound or a ligand is bound to a particular active pocket and it changes the conformation, you immediately know once you excite this laser. So no longer the donor will be able to excite the other thing, so receiver. So you exactly know now, yes, my ligand is bound here and there is a conformational change happened to my protein and so on and so forth. So you can do the photon counting here and you will exactly know at what nanometer difference the conformational changes happen. And like again, designing the experiments for this is little tricky, and you have to exactly orient your molecules in a right fashion. Only then the experiment works. So but yeah, people, SMFRET is relatively very small community, but very helpful community. So people do share their experience and they will teach you out how to do that. And another example here where we have showed immunological synapse in Jacquard T cells. So I said like immuno-oncology applications are also being used as a greater purpose using our microscope. So if you take the synapse of T cells, it looks something like this in a confocal. You'll be not able to understand what is going on the cell, and you'll not be able to resolve it at all. So but when it comes to SMLM on the same slide, if you want to see now, you can appreciate the receptor clusters, whether it is being clustered or whether the individual receptors are present or so on and so forth. So these are the overall applications what I wanted to show you. And for today, I'll be explaining little one or two applications on virus, EVs, and the bacteria here as such. So, but I just wanted to show you the overall picture on a bird's eye view, like what you can do as such. So yeah, so we can classify the microscope users as three categories. Like if you want to do as a molecule quantification or a cell characterization or a tissue characterization, So in case of molecule quantification, you can do particle profiling, whether cargoes loading, unloading, those kind of analysis also you can do. Protein distribution analysis you can do. Then virus, LNPs, and EVs you can do. So in case of cell characterization, receptor quantification, spatial organizations, so spatially you will be able to resolve the questions of nano events, whatever has been happening, or cellular events, what has been happening, or so on and so forth. And if you want to go on tissue level, biomarker clustering, spatial distribution, bispecific or drug deliveries can be seen in case of that. So when it comes to virus, like it requires more of it requires better and accessible tools to unlock the new discoveries or the novel discoveries as such. Because most of them struggle to elucidate the structure or assembly mechanism or host cell interaction and transport and infection and so on and so forth. So I've taken these as few questions, like majorly like who struggles, and we have tried to demonstrate like how it was being easily taken through. So to understand most of them, the go-to for people who are working on virology would be TEM. So sample preparation is difficult. The time slots, what you get is you have to wait for a longer time. The results, what you get is after a very long waiting of 1 month or 2 months. And you'll be able to get really good structure, but you'll not be able to appreciate what are the glycoproteins are present on the surface or host factors which are present, and so on and so forth. But in case of if you have an ONI system, like on your table, you just prepare your samples within 2 to 3 hours, and you just put your slide on it, and you will be able to get your image, and you'll be able to do the analysis. So within a day, you will have your results in hand to characterize such. And these kind of LNP studies, mostly people do in biopharma, like they categorize into LNP1 and LNP2, like they can directly visualize and quantify or classify the virus LNP populations and determine which batch are structurally good, intact, or homogeneous. So if you see class 2 is lesser population and class 1 is higher population, but if you see class 2, it is more homogeneous, and the better population will be always taken up for downstream therapeutic or research application. So these kind of sorting based things can be done. So again, this also is one more classic example where to gain the insights on purity of virus and to measure how homogeneous it is. So two class of LNP were visualized simultaneously here. So one is in red and one is in purple color. So the red population shows a greater heterogeneity here, indicating possible aggregations or very inconsistent kind of a particle assembly. While other purple LNP show very classic, compact, homogeneous, suggesting which has got better structural integrity right now here. And these are the histograms which shows the measurements, and it will quantify these differences for single particle level. So this analysis also will help you to precisely evaluate the purity, homogeneity, and batch quality as such. So, and people who are working on budding, people who are working on these kind of infection studies, also it will be very much useful. So if people generally do TEM imaging again for that, but you will not be able to achieve the budding structure and all using TEM. But in case of DSTOM with super res, you'll be able to do. So it can be easily measurable and compare the distribution of virus with specific labeling. And if you see here, the GP, that is the glycoproteins, have been labeled with a red here. And if you see the pantas here, it allows direct observation of protein clustering. So at the plasma membranes, so where virus-like particles can be seen exactly. So here, the region what I've been showing in the zoom region, so it highlights the localization assembly of hotspots and the budding sites, right? So compared to TEM, so DSTOM not only provides you high spatial resolution, but also molecular specificity. So labeling, quantification, analysis of viral protein distribution, assembly dynamics can be appreciated using this system as such. So understanding one more, it is a classic case where if you want to understand the machinery or assembly or transport of proteins, again, we have helped to solve this here. So helping researchers to understand how how viruses use the cellular and transport pathways for assembly and movements. So here in case of your zoom region, it highlights clearly the nanoscale events or the site of assembly points or viral assembly and intracellular transports. So it demonstrates exactly how virus hijacks the host cell machinery during the infection. So these kind of studies can be used. So as I mentioned before, people who use, there are like 4 or 5 clients who have actually put their system into BSL-3 as a dedicated inside the laminar hood, and this is a real picture which is being taken from the customer site. So even dynamics can be studied. So here we have used influenza virus. We have treated on HEK 293 cells. So you can see that assembly sites are showing slow diffusion patterns here initially. And once it is being assembled, the apical on the apical membrane, it shows a slow moving, but on the basal membrane, it shows fast movement, so representing the very high transport across the cell surface. As once the virus is assembled, then the transport mechanism is faster. So these kind of studies or these kind of mechanisms or these kind of tracking can be done, and this is what exactly the microbiology community would be looking for as such. So apart from this, there is certain other things like uptake study we have already done. We have already seen this. So this is one of the classic case where HPV labeled with 647 and subject to different ultrasound treatments. And this clearly demonstrate as HPV is being labeled, it shows how particle can be directly visualized during the virus entry into the host cell, and also helps the researchers to have virus cell interaction in understanding in the real time. So the imaging reveals more of viral trafficking pathways or entry routes, providing to helps us to understand how it attaches or how it moves or how it internalizes within the cell as such. So this is what we have done. And there's one more study where they have shown that how exactly this virus-like particles can be labeled, and using different ultrasound techniques, they have seen whether it will bind on the surface or it will uptake into the cell or it will aggregate. So these kind of studies people have done using our microscopes. So coming to the bacteria's part, so the major issue is subcellular localizations inside the bacteria cannot be done, or SPT. People struggle to do SPT inside the bacteria. If you want to track a particular molecule, what is happening inside that, or cell wall or membrane studies, if you want to do, people really struggle for that, or effect of antibiotics. Who are working, if they are working on stress proteins and so on and so forth, they can all use this only to resolve their questions as such. So this is one of the study where they have used de-storm technique, and they have stained with OMV with ATO 488. So usually the membrane bacterial outer membrane vesicles, right? So which is represented in green in color. So to show this interactions in bacterial membrane, it is really difficult, or it is I'm sure like people who try to do this on confocal, you would have got images something like this, where you cannot appreciate, or where you will not be able to understand whether your OMVs have been attached to the cell membrane or not. So using SMLM as a technique, you will be really fascinated to see exactly and with more crystal clarity how these OMVs have been interacting with the membranes as such. So there is one more study where Lignolia viruses, bacteria has been stained in the lung here, and we have showed how the host cells are revealing the nano-host pathogen interaction studies. And during the infection, and it also helps to visualize the bacterial infection pathways and understand how this exact virus manipulates the cellular machineries to survive and spread. So this is one publication. I can share a couple of publications later on related to what we have been shown. I mean, there are like more than 44 publications only on bacteria search, which has been noted in all Q1 journals like Cell Science and Nature and PNAS. So I can share you a couple of them after this presentation also. So here there is one more quantification where they have tried to see a transcription factor for an antibiotic resistant mechanism. So novel antibiotic mechanisms are required to overcome the present resistance. So they try to see whether they could achieve it, right? So they have used palm technique here, and they have labeled with Dendra 2. So this is again a transcription factor. So upon the control, you see very less number of transcription factors, but upon the treatment of antibiotic, you see there is a 7-fold change in case of this transcription factor, whatever they were interested to check on. So, and you can also do secretory T T3SS system is what is being assessed in case of this study. So they have used single molecule tracking to understand this. So SPT for bacterial type 3 secretory system, they have tried to see here, and they have tried to visualize the secretory apparatus assembly and protein movements. So they have used a fixed sample here. So they have just used DStorm for here, and they have tried to map and localize the protein on diffusion, behavior, assembly, dynamics, and so you get a lot of information, like each and every thing you can measure. You can draw circles and keep your bacteria of interest, which is clean and neat, and you try to track this particular protein that is TS30 T3SS, and you will exactly know the movements of that, and you will get a lot of information on that. So this is a similar kind of study, but they have used HQF PA M33. That is, here it is a live cell. So that's why they have used PA M cherry here, that they have done the live cell tracking. And what they have tried to see is whether the adoption for the stress can be seen. And HQF, I think it's an chaperone, RNA chaperone, and they wanted to give a stress under various conditions, and they wanted to see how the movements of this HFQ protein has been happening inside E. coli cells. So this gives you a comparison between the control and nitrogen starved cells. So this is your control and 24 hours nitrogen starved cells. It's so real dynamic changes in molecular organizations and forming the stress response of the foci can be clearly measured. So yeah, this is a publication which I've taken that slide from. So this is about a small part of microbial thing, and we have dedicated software and kits. I just wanted to run through this. So we have NemoS, which is a dedicated software which comes with the system, and we have Cody. So we do wanted people to just do the analysis on the scope and waste their time. So we put everything on cloud for analysis. So people have to just use the microscope to acquire it, and the post analysis can be done on cloud, and it is completely free. So any user can register, and it is unlimited data storage. What we give, we don't charge for your data storage, and we don't charge for number of users. It is unlimited and it is free. So that is a good part about our softwares. So you can, this is how the software looks like. For example, I've shown EVs, like you can invite your collaborators here, you can do clustering analysis, counting, batch analysis. So if you have multiple files, you don't want to do analysis on individual, you can just select everything and do a similar kind of batch analysis for everything, and it will be done. So usually most of the people who do SMLM, for every step, there are like this software shows you multiple steps. You try to do step 1, step 2, step 3, step 4, step 5, and your analysis is done, and you can create history, complete page as a report. So to do this, people generally take like 2 to 3 days to analyze one file, because of there are so many normal pipelines. So here we have built in everything as a compact pipeline to give you the analysis result. So this is how the automated analysis reports look like. So, and coming to kits, like we give you DStorm kits. We have DStorm kits if you want to do DStorm application. We have discovery kits in that, and we have LNP analysis kits. We have EV profiler kits. So you don't have to worry about coming into SMLM field and trying to figure out what we have to do and how we have to do. Like we have simplified all that for you. So this is a complete solution, as I said before, like from sample preparation, we have solution. For acquisition, we have solution. For analysis, we have solution. And we have now solutions for automation also. Like you don't have to even perform the experiment. Like we have Apollo Flow, which can do the complete experimentation part also. So I don't want to again go through very deep in case of EVs, because we have already seen a lot of things with EVs. So people try to struggle with EVs, as I have already told, because it's small, heterogeneous population, various biomarker expression would be present and shares properties from the cell. So it is very difficult to show on a standard microscope. But with ONI, we can clearly resolve it, and the readers or people can appreciate it. So we, like, as is told, there are some kits. So how it is being available. So these are the chips what we give. So we give you 3 chips per kit. So 12 samples can be run. And so the sample costing is also very cheap. You don't have to worry. It is not a big cost at all. It is a very small cost what it comes. And you just perform it. Like for EVs, it just take 3 hours for your preparation. And for DStorm, it is just 2 hours for your preparation. And LNPs, it is around 3.5 hours for your preparation. So it is very as simple as that. So you just, how does this kits works? So surface preparation you do, then you capture your EVs, then you stain them with required biomarkers, whatever you want to, you fix it, and just you take images. And once you take images, you just do clustering analysis and EV analysis. We have a complete software solution for that, which pulls out everything for you within minutes. You don't have to put any efforts to do that, and your report will be ready. So we are excited to launch the new scope now. So it is called Apollo Scope, which is the next generation one. So what is the update between Nano Imager and Apollo Scope? Here you cannot do SMFET, unfortunately. But yes, in the case of field view enhancement, you can. We have increased from 5080 to 110 into 110. So that was liquid cooled. This is an air cooled, and we use Hamamatsu Flash 4 camera there, which is having a 85% quantum efficiency. And in case of this, we use Teledyne Prime Plus camera, which has got 95% quantum efficiency. So these are some few gallery pictures people have taken and people have uploaded in our site. So I'm just showing you, and I'm open to any questions, whatever you have. So we can take questions. And if you have any questions, I'll be happy to take few questions. Yeah. So this is the complete suite what we have. So as I said, it is a complete solution what we give. So this is the automated flow, what I was speaking about. It is a fluidic machine which automates things for you. Like you have to purchase kits for that, and it does everything on its own. Yeah. So thank you. Thanks a lot for this opportunity, Dr. Deshmukh sir. And I would be happy to take if there is any questions, or you can drop me email if you have anything, any questions on it. I'll be more than happy to answer it.
Thank you so much, sir, for such an insightful and informative presentation. If anyone has any questions, so we can take questions. If no one has any questions, we can conclude the session. On behalf of the Microbiologist Society India, I would like to sincerely thank Dr. Nagabhishek and his team from Imperial Life Sciences for sharing their valuable knowledge and time with us today. I would also like to thank all the participants for attending and making this sessions successful. Thank you everyone and have a great day.
Thank you. Thanks a lot.
Arya, hello.
Yeah.
Hello. There seems to be few questions in the chat box. Hello.
Yes, sir.
There are few questions in the chat box.
Okay.
So, Arya, give time.
Yes, sir.
Hello.
Yeah. So the software is
Arya,
third party guarantee or warranty or software and any update version in future. To answer this question, the software is our own software. So there is always auto updates. So and it is a cloud based, so you don't have to even update the version of the software. So it automatically gives you the newer version because it is a cloud based enhancement. And we don't charge you as I told, it is an unlimited storage. And as I told you, because the file sizes are really big, you'll be taking more than 10,000 images to stack up and give one super res. So usually it will be like 4 to 8 GB or 10 GB file sizes. So you can do unlimited upload of those huge files, and it is absolutely free of cost for you, and you don't have to worry. And all our analysis pipelines are free of cost. You don't have to pay for even the analysis and pipelines. So yeah, so answering, I think Dr. Subhash had asked that question. I think I've answered that. And Dr. Prakash, sir, how much for sample processing and time and cost of instrument? So to answer you this question, in kit section, I had told you. So in kit section, as I told you, like the sample processing varies, depends upon the type of technique what you're doing. And we provide you kit for DStorm, which usually takes 2 hours. And LNP kits usually takes 3 hours. And we have one more EV kits, which usually takes 3.5 hours to 4 hours to process your sample. And coming to the cost part of the instrument, I think we have our national manager who takes care of commercial aspects. So I think Mr. Deepak Sharma, sir, is there. So he can answer the question regarding the cost, or we can send you the code personally if you can leave the details for us.
I think there is one more question below that.
And how do you achieve molecule specificity by labeling? How do you achieve molecule specific labeling and detection? So you can do by two types. Like as I said, like if you want to do for fixed cell samples or life science samples, you have to decide. And if your molecule of interest has got any specific antibody against the target, you can create a primary antibody for that, and you can use secondary antibody conjugated with fluorochrome to it. So that is the way you will do for fixed samples. And if you want to do on live samples, then you'll do palm technique. So the protein of interest should have downstream attached with PA, GFP or PM cherry. So you will be getting a molecule with which can glow or blink now. So that way you can do your palm technique if you're looking for a live cell. So if I have answered that question, Dr. Prakash.
Yeah, to some extent. But this
Okay,
is
so if you want to know
green
more, yeah.
Now, how you differentiate then green fluorescence protein?
Sorry?
Now, when we do the green fluorescence protein,
Yeah.
we also will be able to localize.
Yeah.
So this method, how it differs than conventional process.
So when you say it is GFP, So normal GFP cannot be used for this. We use a PA GFP, that is a broken form of GFP, a broken form of RFP. So naturally, they have got some deletions in their protein amino acids. So there are few amino acids which is deleted purposefully. So these are not stable GFPs anymore. So automatically, this has got switching on and off property. So that's how we'll be able to do it. So we have to use PA GFP or PA mCherry, something like that.
You do outsourcing also? If we give sample, will you to identify where the target is? Suppose we are working on RNA inhibiting peptide antibiotics, RNA biosynthesis, but we don't know whether it is in RNA polymerase or sigma factor or any other transcription factor.
Okay.
Possible to detect the specific localization or specific target?
To answer the question, yes, you can do it. It is a comprehensive experimental flow, what you have to design to achieve, right? Because you wanted to see multiple organelles or factors to rule out things. So you have to design multiple experiments to rule out and say that this goes here or that is present in this particular factor. But to answer your second part, currently we are not outsourcing it. So it is only the CapEx what we do. So we just sell the instrument. So we don't do any outsourcing currently.
And now you have any service provided in Karnataka, in IISc Bangalore or any other institute?
Currently, no, sir. Like we are having some road shows in upcoming month for in IISc and NCBS. Yes.
Yeah, we can do in Mysore.
So, sir, you're based in Mysore?
Yes, yes.
Okay, okay. I'm based in Bangalore. So probably, sir, which institute are you from?
CFTRI.
CFTRI. Okay, sir. I'll try to come and meet you personally.
Yeah, I am HOD of microbiology department here.
Okay, sir. Perfect. Perfect, sir.
Wonderful. Thank you.
Okay. Thank you, sir.
Yes, again, one more request to you.
Yes, sir.
You prepare one good flyer.
Yeah.
I will upload on our website.
Sure.
And I will circulate also in all groups. Okay?
Sir, information you want me to
Yes.
put in the flyer?
I have a question. Can I?
Yes, yes. Why not?
Yeah. Is there any workshop or training that you hold about the instrument to know about in detail? Kindly let us know if it is possible from your end to organize.
Sure. Sure. So we just had a workshop in AIMS last week for EVs exclusively. We had a hands-on workshop where we demonstrated the instrument. We showed the sample analysis. We did the kit analysis and everything. So it was a very good response. From EV Society, they conducted. They requested us, and we had demonstrated that. So similarly, if you guys, if Microbiology Society wants one such workshop, so we can come up with one such workshop depending upon the availability of the instrument, because we have only one instrument we have currently, and it will be going through Pan India and few Pan Asia. So basically, depending upon the availability of the instrument and people are interested, definitely we can plan one workshop based upon that.
Yeah, so please, I will be happy to host you all. I will be happy to host you all if you can organize in Guwahati University. It is very near to the airport. So nearby institute like IASST, IIT, NIPER, there are few university, other university as well, private and government sector, both.
Yeah.
And even adjoining northeastern state like Shillong, Nehu and all are just 3-hour ride from here. So they also can come. So if you can plan, then I can circulate making a flyer and everything.
Sure, sir.
Yeah, definitely, sir. I'm Deepak Sharma, National Sales Manager from Imperial Life Sciences. So Sagar is also here. So I think Sagar, there is a constraint with us. Like instrument is with us by end of this month, and then most likely it will be again in the month of June last week, right?
Yes. So let me introduce myself. I am Sagar Shah, who's specifically looking after this product portfolio. So, like as rightly mentioned by Dr. Abhishek and Mr. Deepak here, like that we do conduct some workshops, and also we have done it at couple of places, and we have some up lined up in Delhi in the coming week and week after. So, most likely we can do this activity in the month of July or thereafter anytime, because in June it's going to Japan for some other workshops and conferences. So, maybe only in July we'll be having it back in India. So, we'll get back to you, regarding its availability so that we can well organize it. Keeping in mind, I understand, those months would be little harsh due to rains and all, but we'll just figure out some way to organize at a particular center, and we can just call upon the entire society to be part of this workshop. Does
Yes,
that sound
yes,
good?
yes.
Yeah, yeah.
Correct. I
Yeah,
will be
yeah, most
happy.
welcome. July month is very much welcome here. You can organize. Of course, it will be a rainy season here, so there will be issues of flood and all. So that I
Yeah.
cannot say that when you will be fixing, and this. So July is the rainy season here. It is flood prone area,
Okay.
and
No, that's why means
anytime even
we
after
need
that also
to.
is fine. Yeah.
Yeah, yeah. We need to figure out the availability, and everyone should be able to make it up. And it's not necessarily we need to do it for the entire society at one location. If someone is available in South, we can select one center in South. We can select one center in East, and wherever, such centers we can select so that everyone it becomes feasible for them to participate and have an actual hands-on experience. And we will not allow too many participants. We'll keep it little small, like maybe 20 participants, like what we last week we did at Ames Delhi, so that everyone gets hands on the system and they get actual experience about acquiring the data and analyzing it. So that makes it much more I think fruitful.
Correct.
Very, very true, Dr. Deepak sir.
Yes,
What I
sir.
will suggest you, we will locate 4 places in India at 4 corners.
Okay, sir.
And in Guwahati, Dr. Subhash already has agreed.
Okay.
So one will be in Gujarat, one will be in Chennai or Mysore, or some other places we will finalize
Sure,
and
sir.
we will arrange.
Sure, sir. So most likely we'll also come back to you on this, and then accordingly we'll plan that how soon we can do
Yes.
it and how it can be successful for all of us.
Yeah,
Yeah,
it
yeah.
will be
Thank you very much.
successful.
Thank you very much, Arvind sir. Thank you.
Yes, sir. Yes, yes. And it is 100% it will be successful. Many teachers, many researchers are interested to learn the technique, and we will popularize, we will circulate to the every corner of India your flyer.
Sure, sir. Sir, I'll appreciate if you can send your mobile number or something like that, email ID to me on this group, or maybe separately on my mobile number. I have already shared my mobile number on the group.
Yeah, I
Then
have
according.
shared my email ID, my address, and the mobile number in the chat box.
Sure, sir.
Yeah.
Thank you so much. So let's make it a
Yes,
big
yes.
success, and let's work together
Sure,
on. Yeah,
sure, sure, sure.
sure, sir.
Yes. Now,
Any
Arya again express vote of thanks. Arya at the last.
So that was a great session. So on behalf of the Microbiologist Society India, I would like to sincerely thank Dr. Nagabhishek and his team from Imperial Life Sciences for sharing their valuable knowledge and time with us today. I would also like to thank all the participants for attending and making this session successful. So thank you everyone and have a great day.
Thank you, Ms. Arya. Thank you, sir. Everyone have a nice day.
Yeah,
Thank you.
thank you
Thank
so
you.
Thank
much
you
I
all
will
for
for
be
hosting
such
us.
wonderful interactive session.
Thank you.
very
Thank you, everyone. Thank you.
पॉजिटिव चीज का वो hands-on experience बोल रही हूँ